This is Bernard Paulson. I will now go through chapter 10 in this book that describes the construction of a dynamic simulator for glycolysis. First, I would like to orient ourselves a little bit vis-a-vis where we are with this textbook. We have covered part 1 that taught us how to do simulation and how that was applied to simple chemical reactions. In part 2, we started looking at biological features that need to be considered in building simulators. And now in part 3, we will begin to build simulators of particular specific biological phenomena. I should say that this lecture, number 10, and perhaps the last one, number 9, are the most intense and difficult lectures of this entire series. But once, for instance, you're done with lecture 10, that you may listen to more than once, everything else more or less follows what is in this particular lecture. So the outline for today is as follows. First, we're going to look at glycolysis. Glycolysis is a central metabolic pathway at work in most living organisms on the planet at any given point in time. And in fact, if you were able to estimate the amount of material that flows through glycolysis on Earth at any given point in time, you would get a number that is much higher than our entire oil consumption as a point of reference. So this is a really important pathway. The systems description of it contains 20 compounds and 21 reactions. And by system, I mean a network placed inside a system's boundary that is converted into a computational model. And clearly, I'll say more about that as we move along. Glycolysis couples to 3 cofactor pairs that then in turn couple to other processes inside the cell. Glycolysis can couple to NAD and charge it with a pair of electrons. It makes ATP by substrate level phosphorylation, which means that it can make ATP without the presence of oxygen. And then it incorporates an organic phosphate. And this function of glycolysis is often overlooked, but becomes very clear when a system's description of glycolysis is considered. We will formulate the stoichiometric matrix, and I will spend some time talking about its null spaces that contain pathways and pools, as we've seen here and there throughout these lectures. Once the model is defined, or the network is defined, you can load data into it, compute the steady state, and having the steady state allows you to set up a simulation model that you can then use to simulate the responses of glycolysis to perturbations. We'll spend a fair amount of time towards the second third of this lecture to interpret the results from the simulation. We will look at the concentrations, we will look at the fluxes, and I will introduce you to how to form pools and ratios. The latter two cannot be dealt with in detail in one lecture, but as you listen to the series of these lectures, you start to appreciate how pools and ratios are used to interpret the function of biochemical reaction networks from an engineering point of view. All right, so we are now going to build our first biologically meaningful model based on what we have learned in this course so far. And we're going to be looking at glycolysis, and unlike what you might have seen in a biochemistry textbook where you just look at the reactions, we have to form a system description around glycolysis that shows how it interacts with its environment. So glycolysis as an open system is shown in this diagram here. So basically, glucose enters a cell, and we are going to be thinking about here the red blood cell for reasons I'll mention in a moment. And let's imagine that the solid line around the network shown in this diagram is actually the bilipid layer surrounding the cell. So glucose enters, it is a hexose, has 6 carbons. The first few events in glycolysis are such that glucose is charged with 2 high-energy phosphate bonds at the expense of 2 ATPs. The fructose diphosphate that results is split into 2 trioses that then go through the same set of reactions as shown. Towards the end of glycolysis, pyruvate or lactate is then secreted from the cell and goes into the plasma. Inorganic phosphate is incorporated by GAP dehydrogenase. So this is the reaction schema for glycolysis. Again, we are going to consider how this pathway operates in the red blood cell. As we know, the hematocrit of blood is about 40%, which means about 40% of the volume of the blood is red cells. So if you prick a finger and get blood out, 40% of what you get is red cells. They are easily lysed, and based on that lysate, we can do a lot of biochemical experiments. And of course, this was easy to do early on, and this led to the unraveling of metabolism in the red cell. And we have a lot of information about it, and therefore it is a good subject to build models in a course like this one. So glycolysis couples with cellular processes. It couples with purine metabolism, and AMP is formed and degraded in the red cell, as we shall see. We can use glycolysis to deliver redox equivalents as indicated, and of course glycolysis will produce high energy phosphates that can be used to drive cellular processes. This last one in the red cell is mostly used to drive the sodium-potassium pump that in turn is used to balance osmotic pressure and allow the cell to go through volume changes as it circulates through our body. I should also mention that the iron in hemoglobin is functional in the +2 state, but it is constantly being oxidized to the +3 state. So the red cell has to meet that oxidation load, and it does so by providing electrons to iron. So this coupling with cellular processes, it takes place through cofactors, as we have described before, and then the cell has coupling with its environment. It senses, of course, sugar level in blood, and same thing turns out to be true with lactate. As I said, it is easy to get a hold of the red cell, open it up, and measure concentration of metabolites. and some historical data from such measurements is shown here, where we have the concentration of the glycolytic intermediates in millimolar concentration, and we see that glucose and lactate are present in fairly high concentrations, and the rest of them are pretty low. Then we have information about the cofactors and carrier molecules. We see that inorganic phosphate, line 18 in this table, is pretty high, 2.5 millimolar, and ATP is around 1.5, as shown. The biochemical reactions that take place are also well known, and here are the 21 we need to be concerned with. We have the glycolytic reactions themselves, the 11 reactions there. We have some AMP metabolism that we have been introduced to earlier. We have exchange reactions of the primary products of glycolysis. We have the two loads, the energy and the redox load. We have the imports or the boundary fluxes. And as we did in chapter 8, we're going to consider these to be constant and in this chapter, and in a way these are therefore forced on the cell. And then of course the red cell, like all cells, exchange protons and water with their environment. I will note here that the import of phosphate is not explicitly accounted in here. And the input and the output of phosphate is hard coupled, the way this model is formulated. And the consequences of that are described in a homework set that you can go and solve as a variation of the model described in this lecture. So that is a description of the biochemistry and some of the physiological roles glycolysis plays in a cell like the red blood cell. Based on these reactions and compounds, we can formulate the stoichiometric matrix for this system, and that is shown here. We have the glycolytic reactions, AMP metabolism, the export of pyruvate and lactate, and so forth as columns. And then we have the compounds ordered in this matrix the same way as I had them in the table before. The dimensions of this matrix are 20 rows, 21 columns. The rank is 18. So there are 18 independent rows or independent columns in this matrix. And we mean by that, of course, linearly independent. Given the fact that the rank is 18, dimension of the null space is 21. The number of fluxes is 21. We compute the dimension of the null space as 21 minus 18, or 3. And we'll show that these three dimensions in the null space represent pathways. So the null space of the stoichiometric matrix here is spanned by 3 pathways. There are 2 dimensions in the left null space as shown, and this means that we have 2 conserved quantities, and these we will refer to as hard pools later on because they cannot change in their size at all. I want to mention that we have to perform quality control on this stoichiometric matrix, and this slide illustrates elemental balancing. So every column in S sub I, which is a reaction vector, represents a chemical transformation. And for it to be chemically accurate, the reaction has to be elementally balanced and charge balanced. And I presume that most people listening to these lectures have had general chemistry and have gone through elemental and charge balancing of chemical reactions. This balancing can be done with one simple matrix multiplication, as is detailed in the companion book to the one used in this class. And I'll just briefly illustrate that here. First, one forms an elemental matrix, And this is a very simple accounting matrix that shows the composition of every compound in your network. And it's organized by forming 6 rows that corresponds to the 6 elements found in organic molecules. And then every column in this matrix is a compound. In here we show 2 of them, and the entries for the column for water will be 2 in the row for hydrogen and 1 for oxygen and 0 everywhere else. So this column represents the elemental composition of water. The second column shown in here is glucose that has 6 carbons, 12 hydrogens, and 6 oxygens. And you see the corresponding entries in this column. And then there is 0 on nitrogen, phosphate, and sulfur because that is not found in glucose. Those elements are not found in glucose. This we can do for every compound that is found in our network, and it's easy to look up the elemental composition of compounds. We can form this matrix E then readily for our network of interest. And to elementally balance the stoichiometric matrix, we need to multiply the elemental matrix from the left, and that should give us 0 for all the columns in S that are elementally balanced. For instance, you can readily see that if you take, for instance, the row for carbon, and you take that row in E and multiply it by a column in S, you will get positive values of all the carbons that are being made in the reaction, and a disappearance of all the carbon atoms in the reactants in this reaction. Here is a simple example that shows how this works, where we just consider 2 elements, oxygen and hydrogen. I have 3 compounds, hydrogen, oxygen, and water. The first column in the elemental matrix has 0 and 2, so that's hydrogen. The second has 2 and 0, so that's oxygen. And the last one has 1 and 2, so that's hydrogen. The stoichiometric matrix for this system is just a column. And you see it has -2, -1, and 2 in it. That are simply the stoichiometric coefficients in that reaction. And if I multiply this column vector from the left with the elemental matrix, I get 0, which tells me that this reaction is elementally balanced, which is self-evident from the reaction written. Sometimes elemental and charge balancing can be a little more intricate, and therefore the fact that we can do this with a simple matrix multiplication is very helpful. The upshot of this product being 0 is that all the rows of E are actually in the left null space of S. That is simply a mathematical statement. We can now take the stoichiometric matrix that we had 2 slides ago and annotate it with various information. So the stoichiometric matrix we just looked at 2 slides back is found within this dashed box that I show here. The elemental balancing is shown in this solid box, and you see 0 for all the entries for all the elements in the glycolytic reactions. For the exchange reactions, you do not see zeros, and the reason is that there is in the exchange reactions there is only an internal compound, no external compound, and each one of those reactions represents a net gain of elements going into the cell or loss of elements leaving the cell. The next block in this matrix are the pathway vectors. If they are put into a column format to form the pathway matrix, shown here as capital P, if I multiply S times that pathway matrix, I'm going to get 0. This is simply a statement of the fact that every pathway is in a null space of S. We'll look at these pictorially in a moment. There are other things you can calculate, like the connectivity. That is just the summation of all the non-zero elements across a row in the stoichiometric matrix, and that therefore tells you how many reactions a compound participates in. If you look at the rows for ADP and ATP, you see that they sum up to 6. Water participates in 3 reactions here, and proton in 8 of them. You can also calculate what's called the participation number, which is simply a summation of the non-zero elements in a column, and that tells you how many compounds participate in that particular reaction. I will ask you to think about how you can show a pathway is elementally balanced. This simply means that the elements going into a pathway by the uptake reaction are completely balanced by those that leave at the other end of the pathway. Now let's look at these basis vectors that I have picked for the null space. I can show them pictorially. The first one represents glucose coming in and leaving as 2 lactate, and in the process making 2 ATP molecules that can meet the ATP load placed on the cell. The second pathway is the lactate pyruvate exchange that involves the uptake of lactate, secretion of pyruvate, and the net production of 1 NADH that can be used to meet a load. And the third pathway is a little bit of an artificial pathway which just imports AMP and then secretes it again. We will see later how this pathway breaks into more complicated biochemistry later on as we consider the purine metabolism and its totality in the cell. These pictures, these pathway maps correspond to the vectors shown at the bottom of this slide, and these are the same pathways as I had on the previous slide. You can trace the entries in the rows for the pathways to the map, and I'm going to pick one of them here to illustrate that. Let's look at the second pathway. It has a few non-zero entries in it. There it has a -1 on lactate dehydrogenase because that's run in the reverse direction. There is pyruvate secretion leaving. There is lactate secretion with a negative number, so it's coming in. There is the load that I can meet, which is 1 NADH, and this particular reaction, this particular pathway actually produces a net of 2 hydrogens. All right, so that is how we describe the null space. And you can, if you think about it for a while, in your mind, just graphically add up these icons on top to get any flux map of interest. Now this is a 3-dimensional null space with the flux vector in it. So if I measure 3 fluxes independently, I can specify where I am in that space. And in this case, the measurement of glucose uptake will specify the first pathway. The secretion of pyruvate will specify the second pathway. And for the third one, I'm going to use a number of 0.014, I think it is, that will become apparent later on, whose origin will become apparent later on. So I can just add up the 3 pathways in here in the ratios given by the measurements to compute the steady state. And that is shown in this slide. The uptake rate of the red cell in homeostatic state is about 1.12 millimoles per hour. So through upper glycolysis, we have 1.12. Then we have the split into 2 3-carbon units, and the flux doubles. Then you see the difference between pyruvate kinase and lactate dehydrogenase, which is 0.224, and that corresponds to the pyruvate secretion and so forth. You can walk your way through this flux map by looking at these numbers or the bar chart to the left, to the right, I should say. I similarly show here the steady state concentrations that are measured in that steady state. And I just note here that the distribution of these values numerically is much broader than that for the fluxes. For instance, entry 18, compound 18 is phosphate at 2.5 millimolar, compound 17 is ATP at 1.6, 12 is lactate at 1.36, and the first one is glucose at 1. Everything else is relatively small. I should say that the concentration of water is arbitrarily put to 1 because it doesn't influence any reaction rates, as we'll see later on. So I now have a full definition of the steady state. I know the flux map and I know the concentrations. If I now consider mass action kinetics for all the reactions, I can, based on this information, calculate what's called a perk, or a pseudo-elementary rate constant that is consistent with the steady state that I have measured. And this is very important because we can simply calculate the approximate rate constants under the condition where we are observing the cell. Here is the mass action ratio that I discussed earlier in the class in lecture 2, and that is the same, has the same definition as the equilibrium constant, except it is computed with the steady state values and not the equilibrium values. So in the last column, I ratio the two to get a sense for how far from equilibrium the reactions are in a steady state. The three reactions highlighted here are hexokinase, phosphofructokinase, and pyruvate kinase. And these are known as the irreversible reactions in glycolysis. And you see they all show up with 0 on this scale, which means that they are far, far from equilibrium. Contrast that to these three reactions shown here, where this ratio is very close to unity, which in turn means that these reactions are very close to an equilibrium state in the steady state of the cell. And you can see that one of the reasons for that is that these rate constants, these perks that correspond to these reactions, are very large and have very fast dynamics. And now you can recall the example we did in chapter 6 that says that when you have fast internal dynamics relative to the exchange rates, you tend to get reactions that are close to a quasi-equilibrium state in the steady state. So, having computed this table, I am able now to fully define my model. I have my concentration vector, I have my flux vector, I have my stoichiometric matrix, and I've calculated all the kinetic constants based on data, namely measured uptake rates and measured concentrations of the compounds inside the cell. I am therefore ready for simulation. I, of course, have to pick now the initial conditions or the perturbation that I'm interested in, and then I can just numerically solve. What I'm going to do now is to go through that process using the same perturbation as we have been looking at for basically simpler prototypic version of this system that I have now described in its entirety. So the simulation we are after is this 50% increase in the rate of use of ATP. the cell all of a sudden has to use ATP faster. And this is now a boundary flux as before, where the load is, the ATP load is a kinetic constant times the concentration of ATP, and we are going to bump this value by 50% at time 0. We are then going to look at some of the characteristics of the response. First, I want to point out that if you look at the perks in the table that I had up a couple of slides ago, I can estimate what the range of response times is going to be, and I decipher that there will potentially be some fast events, very fast events associated with these quasi-equilibrations that might be activated, but most likely I'm going to see a response on the order of a few hours and then on the order of a few tens of hours. The pools and the transitions will be discussed later on. So basically what I do is I bump up that parameter by 50% at time 0, and that'll imbalance this one flux exactly at time 0, or time 0 plus as we call it. And you see that that flux has gone up 50%. I can then solve the equations as we've talked about and plot the concentrations as a function of time. I have selected just a few of them here for illustrative purposes. I'm showing ATP, ADP and AMP and glucose in this graph. You see that on the very fast time scale, not much is happening. On the time scale of order of hours, I get the concentrations wiggling around. And then if I expand the time scales to 50, I see that those wiggles are now, right up front to the left of that graph. And then there's a longer, slower time scale on which you can observe motion, like you see AMP dropping mildly on that time scale. Note in the table that is shown there, the final flux state at T50, where you're essentially back into the steady state, is the same as the initial state right before the perturbation. This is because the flux map has to return to the same steady state since I fixed the input boundary fluxes. We discussed this at the end of chapter 8. On the other hand, if you look at the concentrations at time 0 and time 50, they vary. They will have to be readjusted relative to the new rate constant on the ATP load, but still the flux map in the end is the same. And this is what I meant when I stated earlier that when you look at systems, the fluxes are the dominant variables and the concentrations are subordinate variables. They have to behave to meet the needs of the system as a whole. And of course, the kinetic constants are genetically determined variables. Now, I can prepare my array of phase portraits as we discussed in chapter 3. Here I'm showing all the phase portraits for the fluxes on the time scale of 0 to 10 hours. And I can now look at this diagram to get a first pass impression of what the dynamics are like. My first observation is that there are 3 fluxes in here that show particularly strong dynamic dimensionality. That is the AMP secretion rate, the pyruvate secretion rate, and the ATP load. The phase planes of those fluxes plotted relative to one another are of particular interest, as we'll talk about later. As I look through this matrix and look at other phase portraits, I see that they all more or less look the same, that indicate that these fluxes are behaving pretty similarly, And this again speaks to the effective lower dynamic dimensionality of the system on a longer time scale. If I look at my final time scale, above 10 hours, you see that these phase portraits of the fluxes have simplified even further. Okay, I can now look in more detail at the particular fluxes that I picked out and zoom in on those particular phase portraits. The first one in panel A here shows the ATP load plotted versus the AMP secretion rate. At time 0, I'm at the point shown at what is that number, 2.24 of ATP, and that AMP input rate at 0.14. At time 0, I instantly move the ATP consumption rate out to the point 3.36 and then watch the response. And you see that the load of ATP drops and the AMP secretion rate goes up, and then that turns around, moves down vertically, and eventually goes to the starting point. On the plot below, I show the pyruvate ATP secretion, the pyruvate secretion rate versus the ATP load. And the initial reaction here, after the sudden move of the ATP load of pyruvate secretion, is straight up vertically as pyruvate is now being secreted, and then that turns around and eventually settles down in the original steady state. The reason for that sudden increase I'll talk about in a moment. And then we can plot also the pyruvate secretion rate versus the AMP secretion rate and look at that trace in light of what I just talked about. This slide shows on top the three-phase portraits I just talked about. The pyruvate secretion rate going up is a result of the sudden draw on ATP and the acceleration of pyruvate kinase. That leads to the secretion of pyruvate. And since glycolysis has two dehydrogenases in it that tend to be balanced, the gap dehydrogenase and the lactate dehydrogenase, the secretion rate of pyruvate will imbalance the redox pool a little bit, and that's shown in panel D and E in this diagram. Panel F shows the kinases at the beginning and end of glycolysis. As you see, the initial reaction here is a drop in the flux through hexokinase and an increase through pyruvate kinase, and the reason for this is the drop in ATP concentration and the increase in ADP concentration that takes place as a result of the perturbation in the load parameter. Before I leave this slide, I should note that the axes on panel C are mislabeled. These should be the opposite. The pyruvate secretion rate should be on the y-axis and AMP secretion rate on the x-axis. I can also look at the phase portraits for the concentrations, and I can observe that these are also effectively low dimensional. But there's a lot of motion in these, and I will describe later on how these are all pulled together to simplify our understanding of the dynamics of the concentration vector. And here we see the concentration changing on the longest time scale, and we see the vast simplification that is taking place in these responses on this time scale. Okay, so at this point we've been able to formulate the model based on data, find the steady state, and simulate. We can do a first pass analysis or interpretation of the response. We have 20 concentrations to look at and 21 fluxes to consider. And of course this is complicated, but so far we've been able to decipher that the dimensionality of the dynamic response is actually much simpler than the dimensions of the stoichiometric matrix indicate. To help further understand the nature of this response, we are going to look now at the structure of the system, the structure of the network, the structure of the biochemistry. And this is going to move us towards what we think of as systems biology, because the concentrations aren't all independent, or the fluxes, but they are interrelated. I want to start by showing you this image of the glycolytic system that we have looked at earlier. I am not using compound names here, but I'm using the icons that were introduced in chapter 8. You see the AMP carrier is represented by a square, and the high energy bonds on it are represented by the circles. Glucose looks like a bow tie. It's a hexose made up of 3 trioses. It is imported, and you can see phosphorylated twice at at each end of these 2 attached trioses. They are then split in 2, and then at the GAP-DH reaction, the inorganic phosphate that comes from the outside is incorporated, and eventually that inorganic phosphate that's incorporated moves out of glycolysis onto ATP in the pyruvate kinase reaction that I then consider to meet the load. So that open circle moves into the system from the right at GAP dehydrogenase and comes out on the left where that load parameter is. I've also shaded the compounds or left them white depending on the redox state. So all the reactions before GAP dehydrogenase have compounds that are shaded, and then you see that shade that represents the redox potential is moved onto NADH in the GAP dehydrogenase reaction. So all the glycolytic intermediates past that state, past that reaction, are now white. If you go all the way down to lactate, the pyruvate lactate reaction catalyzed by lactate dehydrogenase, I show how the shaded form of the NAD cofactor, the one that has the redox potential on it, exchanges it with pyruvate, and now lactate is shaded as it carries a redox value to the cell. So if you think about pathway number 2, if I reverse the lactate exchange and take that shaded lactate in, convert it to pyruvate, and secrete the pyruvate now being white, I will have produced one reduced molecule of the NADH carrier system. So this is a way to visualize the flow of material, energy, and redox potential in this system pictorially. Based on these considerations, I'm going to argue now for the formation of pools or aggregates of the compounds that mean something to us. I'm going to start with the trafficking of redox potential. So glucose has the has the potential, I should say, to make two NADHs in this network. Glucose would come in, be broken into two GAP molecules, each one of which can generate an NADH. Then, if you move past the GAP dehydrogenase reaction, you look at 1,3-DPG and so forth, all the way down to pyruvate. There are no redox exchanges, so all these compounds have no value to the cell from a redox standpoint. Then, of course, at the end, here, when pyruvate is converted to lactate, I dispense an NADH and actually endow lactate with a redox potential and secrete it. I have shown some flux values on here that are of interest. So if I take 1.5 flux units of glucose in here, I would have 3 flux units going into GAP dehydrogenase. And if I have 1 flux unit of load on this cofactor, that will then lead to downstream the secretion of 2 lactates and 1 pyruvate. Because I'm only utilizing one of the 3 NADHs that can be made, given that input flux of glucose. So you see that indirectly one can measure the oxidation load on this cofactor by measuring the relative secretion rates of lactate and pyruvate. So I basically, by drawing this out, formed pools that tell me the redox value of various metabolites in the cell. I can do the same thing with the ATP value of every intermediate in the system. The number with the parenthesis above the compounds in this diagram indicate the high energy bond value of the intermediate. So glucose, as we talked about earlier, has the potential to make 2 ATP molecules. After it's imported, it gets phosphorylated once to form the hexose phosphates. And since I've spent an ATP on glucose, these hexose phosphates are now have a value of 3 ATPs, or 3 high-energy phosphate bonds. I load a second ATP molecule by using phosphofructokinase to basically make fructose bisphosphate that will now have a value of 4 high-energy phosphate bonds. And as I showed earlier, that is split into 2 triose phosphates that now each is valued at 2 high-energy phosphate bonds. The phosphoglycerate kinase extracts one of them, and you form the phosphoglycerates that now have the potential to make only 1, and the second ATP is then extracted by pyruvate kinase to make pyruvate. And pyruvate and lactate have no high energy phosphate bond value in the system and are secreted. This particular image is calibrated in flux values relative to 1 input of glucose. And 1 input of glucose leads to the formation of 2 ATP molecules and is silent on whether pyruvate or lactate is secreted. The third cofactor coupling in glycolysis is a little more intricate and takes a little more time to think about. The first one I've already alluded to, and that is the fact that the inorganic phosphate incorporated with GAP dehydrogenase eventually travels out of the system and is used by ATP to meet the demand, or as shown in this diagram, is moved onto a molecule C that now is endowed with a high energy phosphate bond. This is a motion through the system and represents the assimilation of an inorganic phosphate. Now there is also a cycle of phosphate groups in glycolysis, and that's illustrated here. For every glucose molecule going in, there are two ATP molecules used to charge the intermediate. It's then broken in two, and then at the gap, at the hydrogenase stage, at the phosphoglycerate kinase stage, two are extracted, and you can view them as being cycled back and used to charge the next glucose molecule. Whereas further down in the reaction, as shown on the upper panel, there will be two high-energy phosphate bonds that can be moved through the system from the two molecules made in pyruvate kinase. So you can think of phosphates cycling around in glycolysis, hopping off and on the glycolytic intermediates, and vice versa for the adenosine phosphates, whereas there is a trafficking of inorganic phosphate in and out of the system. And that is, of course, the net effect of the pathway. Based on these considerations, we can now define pools and ratios that are meaningful from a physiological standpoint. And I'm now thinking about the metabolic physiology of this system we have defined. We can define pools that are charged or occupied with a property, like a redox equivalent, and a pool that is vacant, but could be carrying that property. And if I add those two up, I get the total size of a pool or the total capacity to carry a property. The ratio of the occupied state to the total we can think of as a charge. And the two examples are given at the bottom of the slide. One is the redox charge of the NADH cofactor. So that is the NADH concentration over the total. And below that is a little more complicated ratio, the one we have been introduced to earlier, the energy charge. And this is a little more complicated because ATP carries two high-energy phosphate bonds and ATP one. The vacancy state in this case, shown in the denominator in the second pair of square brackets, is the vacant spot in ATP plus the two vacant spots in AMP. Then, of course, I can sum things up in the denominator and get the form of the energy charge that we had talked about in chapter 2 as well as in chapter 8. So based on all the considerations that I went to over the last few minutes about the structural properties of glycolysis, we can now form a pooling matrix. And there are few key pools that you can formulate here. The first one shown here is the occupancy of the glycolytic intermediates with potential high-energy phosphate bonds. And as we talked about, there are two of them in glucose, 3 in the hexose phosphates, 4 in fructose diphosphate, or fructose bisphosphate, as it's sometimes called, 2 in the trioses, 1 in the phosphoglycerates, and of course 0 in pyruvate and lactate. So the ground state of the glycolytic intermediates with respect to energy would be pyruvate and lactate. Here I am showing the high-energy phosphate bonds on the adenosines, and this is something we are familiar with. There are 2 on ATP, 1 on ADP, 0 on AMP, and that is mirrored by the vacancy state as I spoke to on the slide or two ago, where that shows that there are 2 vacancy spots on AMP, 1 on ATP, and none on ATP. Similarly, we can write out the redox state of the glycolytic intermediates. All the hexoses can make 2 NADHs. The following 2 intermediates dihydroxyacetate phosphate or acetone phosphate, and the gap can make 1. And then there's a stretch of them that can make none, and of course lactate can make 1, as we mentioned earlier. The ground state for this property is found in these intermediates that have no ability to carry redox equivalents in this system. Finally, I mentioned the pools in the left null space. These are the total phosphate inventory, that is a constant in this case, and the total amount of the NAD carrier, that is only found in 2 forms as highlighted with the red box. These are the hard pools introduced earlier that are found in the left null space. Their amount never changes in time. The first one of these, the phosphate balance, is a little bit artificial because I'm not considering an explicit import reaction for phosphate, as I mentioned earlier. That is delegated to the reader in one of the homework sets. Okay. I can take the pooling matrix and multiply it times the concentration vector as shown on the previous slide and write the pools out. And that is what's done here. Pool 5 is the redox inventory or the number of redox equivalents on all the glycolytic intermediates. Pool 6 is the corresponding ground state, so they are the oxidized form of them. And then I can actually form a ratio between P5 and the summation of P5 and 6 and define the redox charge on glycolysis. I'm going to call that ratio number 3, and that's defined by equation 10.12. At the bottom of the slide, I showed the more familiar charge for NADH. So that's simply the NADH concentration divided by the total. I can now describe the redox trafficking in this network by these 2 charges. The charge can either be in glycolysis or on this cofactor. And of course, they're constantly exchanging them. I can do something similar for the energy state of the cell. So here is the energetic value of all the glycolytic intermediates as described. Here is the ground state. These are the oxidized versions. And then I can form my energy charge for glycolysis as this ratio P1 over the summation of P1 plus P2. And I'm going to call that ratio number 1. And it is analogous to the energy charge on that open square that I had earlier, the AMP carrier. So that's my second ratio. So now I have 4 ratios, that 2 of which describe the redox trafficking and state, and 1 and 2 that describe the energy state. And then finally, I'm going to define the 5th ratio here by ratioing the amount of the incorporated phosphate groups as defined earlier, and the recycled phosphate group, and find the ratio of the phosphate in the cell that's in the recycle versus the total. That should give me a feeling for or a measure of how many of the phosphates in the system are traveling through it versus those that are simply being recycled in there. So these considerations allow me to define 5 key ratios that are linked to the redox, energy, and phosphate trafficking in glycolysis. What I can do, if I want to, is to figure out exactly which fluxes fill and drain the pools that I have defined. And this is done by multiplying the dynamic mass balance from the left with the pooling matrix as shown in here. I now have the derivatives of the pools, and that's equal to the pooling matrix times the stoichiometric matrix. And this product shows exactly how the different pools are being moved by the fluxes. For the hard pools, the entries in this matrix P times S will be all zeros because there are no fluxes filling or draining those pools. They're hard pools. In contrast, for instance, I can look at the adenosine phosphate pools, the charged and discharged ones, and you can see several of the columns have -1 and 1 in those two rows that just represent the exchange between these two. And then there are the two fluxes highlighted by circles that fill and drain the the AP negative pool, the vacancy pool itself. Based on these considerations, I can now draw a pool map that shows the transitions between these pools and how they are driven by the different fluxes. And I can indicate the pool size by the area of the square that I'm drawing. So you can see, for instance, the AP plus pool is pretty large. The P plus pool is also very large. And you can see at the top of this diagram that the NAD pool, NAD and NADH pools are very small in comparison. We introduced these type of diagrams or this type of graphical representation in chapter 8. So this is a very functional diagram of glycolysis with respect to what this metabolism is achieving from a systems point of view. And what I can do now is, as I've indicated earlier, I can now post-process the solution as described in chapter 3 and form the pools just defined from the time profiles of the individual concentrations. And now you can see how the different pools are moved around in red cell metabolism. And you will note that the time behavior of the adenosine phosphates is not too dissimilar from that observed in the simplified versions of this system in chapter 8. The ratios are helpful to help us understand the physiological responses and state of a cell. So here are the 5 key ratios that I've defined, and I can so here they are defined. I can show them also graphically like the pools, and this was a representation also introduced in chapter 8, where I have the glycolytic redox charge, I have the glycolytic phosphate charge, I have the energy charge, and on the very top I have the redox charge on NADH. And this particular stimulus that we simulated here was a perturbation on the rate of use of the high energy phosphate bond. And like we said, that will not influence the redox state of the cell. But in the two steady states, the beginning and the end, but you can see that there is a little bit of wiggling of this charge based on the pyruvate secretion rate that I spoke of earlier. And the other panel maybe worth highlighting in here is the energy charge that dips down, comes back up, wiggles once, and recovers to a steady state that is not quite the same as where we started. This is also a similar response as we saw from the stoichiometric representation of the prototypic pathway at the end of chapter 8. So that concludes the description of the simulation model and its dynamic responses, and I would say an introduction to pooling in a physiologically meaningful network model and the formation of ratios that can be used to interpret the physiological state of this metabolic network. Just a couple of comments about the assumptions. In this model, we are not assuming that any DH is being produced or consumed and is therefore a constant. I believe that the red cell is not known to synthesize this cofactor, at least with the present state of knowledge. The total phosphate conservation is a bit artificial here, as I have mentioned twice in the lecture. The second assumption worth noting is that the forcing functions, the two boundary fluxes for glucose and AMP, are constants. In a later chapter, chapter 12, we will relax the constancy of the AMP input and look at the metabolism that makes AMP. But throughout this text, I'm going to consider the glucose uptake rate to be forced on the cell, although in more realistic models of cells, the uptake rate of a substrate like glucose is regulated by some of the internal variables in the cell. We are assuming that the environment is constant in terms of its pyruvate and lactate concentrations in plasma and the pH. The plasma concentrations appear in the rate laws, as you saw in the tables earlier. And of course, these can vary. For instance, under heavy exercise, anaerobic exercise, you will see an elevation in the lactate concentration in plasma. That makes it harder for the red cells to secrete the lactate against a more challenging concentration gradient. As mentioned in chapter 2, we simulate these processes on a constant volume, and we have here not kept track of charge trafficking in and out of the cell. I also mentioned earlier in this lecture that the ATP is used to drive the sodium-potassium pump primarily in red cells, and the pumping of sodium and potassium helps the cell regulate its volume and its osmotic pressure. These processes are ignored in here. And finally, we do not have any regulation included in this version of the model, and we are simply considering pseudo-elementary mass action kinetics governing the different reactions. We will address this assumption in part 4, where we show how the regulatory enzymes in these pathways, or in this pathway in particular, glycolysis, can be incorporated. So the model that we have built now can be viewed as a model of the mass action scaffold in glycolysis. Any regulation that we build on top of that will bias the mass action trends. And this is, of course, something we discussed in some detail in chapter 9. So we have finally come to the end of this long, detailed, and information-rich chapter. And as I said at the beginning, you may have to listen to it more than once and try to keep track of the details on the slides better when you listen to it again. So what are the summary lessons here, or the summary bullet points? First, we can get draft dynamic models using measured concentrations, using elementary reaction rates, and the associated mass action kinetics. And of course, we also need the exchange fluxes to calculate the steady state flux map. With a model like this, we can simulate the dynamic responses for perturbations in boundary fluxes. And the one we examined in here was a perturbation in the rate of ATP usage. The first draft, the model built here can be considered as a first draft to the mass action scaffold of this pathway. and we can now build around it more processes to increase the complexity of the model and make it more realistic in terms of the physiological processes it describes. In the 2 subsequent lectures, we will show how additional pathways can be incorporated in the model that interact with glycolysis, and we will look at the incorporation of regulatory enzymes in chapter 14. These comments point out the fact that complex dynamic models are built module by module. You build a module, test it, make sure it's consistent with the data that goes into it, quality control it in various ways as mentioned throughout the lecture. You then have another module that you do the same thing to, and then you combine them. This approach here is very similar to the synthetic approach that characterizes engineering design. Building things component by component, module by module, and building them into increasingly complicated representations. In here, of course, we are doing everything in silico. Often in real engineering, you prototype and build things that are more and more complicated as your project progresses. A few bullet points on the interpretation part. I would say the discussion of pools and ratios given in this lecture are introductory, and the way you pool and form ratios in models that you later will build is up to you. The outline given in here was driven by the biochemical considerations, the structure of the network, and considerations of the physiological role that the pathway plays in the cell based on its coupling with the external and internal environment of the cell. So a metabolic map can be analyzed for its stoichiometric texture, and one can assess the cofactor coupling functions of the pathway. Such breakdown of the biochemistry helps define the pools that one considered to be physiologically meaningful from a metabolic perspective. The raw output of the simulation, namely the concentrations as a function of time, can be post-processed with the pooling matrix you form, and that allows you to graph the dynamic responses of the pools and the ratios that you have defined. You can also graph the net rates of reactions that fill and drain a pool, and look at that graphical representation. And I should say that there are many interesting features built into the topological structure of a model. As you can see in the responses given in here, the system naturally overcomes the perturbation that is given to it here by rebuilding the energy charge. And finally, as I just mentioned, we can actually look at a flux map that is based on the pools rather than the individual concentrations. So this is a long and difficult lecture in terms of content. I hope that you are able to get through these concepts as you listen to the lecture, or may have to do it again. And they'll certainly be brought into focus even more in the subsequent lectures when we start implementing the module by module approach that will culminate in a fairly comprehensive description of the core metabolism in the human red blood cell. Thank you for your attention.